Good morning, everyone. Thank you, Professor Mathilde Touvier, for your wonderful presentation and also for organizing this really fabulous symposium. It's really a pleasure and a privilege for me to speak at this symposium. I'm going to give a quick overview on nutrition epidemiology of chronic diseases in the multi-omics era. So as all of you know, in the last several decades, there have been remarkable advances in omics technologies, from genomics to transcriptomics, proteomics, metabolomics, and metagenomics. Those advances, I think, have been extremely important for epidemiological studies, for nutrition in particular. And one of the major implications of those technologies is that it's not just improve the credibility of science, but also allow us to conduct large epidemiological studies using those technologies, because the cost of doing this type of analysis has dramatically decreased in the last several decades. As an example, in year 2000, it cost $100 million to just sequence one human genome. And today, it costs only a few $100s. This is just unbelievable. This has happened within only 2 to 3 decades. And the advances in those omics technologies have really transformed how we conduct nutrition epidemiological studies. Basically, this has shifted paradigm from the so-called black box epidemiology to systems epidemiology. So what is black box epidemiology? It's a type of chronic disease epidemiology that we typically conduct in, I guess, in the 1970s or 1980s, when we look at, for example, the relationship between cigarette smoking or fruits and vegetables and the risk of cancers. We observe some associations, but we really don't understand the biological mechanisms underlying those associations. But today, with the availability and accessibility of those omics technologies, we will be able to incorporate the systems biology tools, such as omics technologies, into large cohort studies and the nutrition intervention studies. So this approach allows us to illuminate biological mechanisms which can enhance the interpretation of causal relationship between diet and the chronic diseases, and also help us identify novel exposure and disease biomarkers, improve disease predictions, identify high-risk individuals and populations for personalized or precision nutrition interventions. Diet is central to all this omics data, because we know that dietary factors can influence our metabolome, our microbiome, epigenome, and the proteome, and also diet can modulate the effects of our genes on risk of chronic diseases. The system epidemiology approach requires us to integrate population, laboratory, analytic, and translational approaches to study the risk of chronic diseases. So the populations can include birth cohorts, observational cohorts, dietary intervention studies. Laboratory approaches have expanded from traditional biomarker discovery to functional studies and the multi-omics analysis. And data analytical approaches have also expanded from traditional risk factor epidemiology to high-dimensional data analysis and the network analysis. But eventually, all those findings need to be translated into clinical predictions, dietary guidelines, and nutrition policies. That's the ultimate goal of our studies. At Harvard School of Public Health and the Brigham Women's Hospital, we have been conducting several large cohort studies to examine the relationship between diet and lifestyle factors and the risk of chronic disease. So those cohorts include the Nurses' Health Study, which started in 1976, Nurses' Health Study 2, which started in 1989, and Nurses Health Studies 3, which is still ongoing. And then the participants in the GOTS cohorts are the children of Nurses Health Study 2, and the health professional follow-up study is a parallel cohort of male health professionals. So in those cohorts, we have been collecting detailed data on diet and lifestyle on a regular basis every 2 to 4 years. We have also collected GIS data and also digital phenotyping data using wearable devices. And our biorepository has stored a massive number of biospecimen samples, including blood, tumor tissues, urine samples, even toenails. And we have already collected more than 20,000 stool samples. We also have a large number of mammograms. So the unique features of those cohorts is the repeated measures of diet, which is very important because that can help us to reduce the measurement errors or the noises in typical dietary assessment, and the long-term follow-up, and a very high rate of follow-up. So far, our follow-up rate has been around 90%. And also multi-generational data allow us to examine the role of maternal diet on offspring's health outcomes. And again, the high-dimensional data will allow us to examine biological mechanisms or biological pathways underlying the relationship between diet and the chronic disease risk. I just want to briefly highlight some of the findings from our large cohort studies. In the 1980s and 1990s, the low-fat diet recommendation was dominating dietary guidelines, public health policies. But at that time, there was very little data on the quality of fat and the risk of chronic diseases. In this study published in 1997, we found that the quality of fat or the type of fat is actually more important than total amount of fat in the diet. So as you can see, that trans fat is the worst type of fat, but saturated fat is also associated with increased risk of heart disease. On the other hand, higher consumption of unsaturated fat, especially polyunsaturated fat from nuts, from vegetable oils, from fish, is actually beneficial. So the take-home message from this analysis is that reducing total amount of fat is not science-based approach because the type of fat is much more important than total amount of fat. And this has very important public health recommendations because at that time, the low-fat diet has led to a dramatic increase in consumption of refined carbohydrates and added sugar. And this type of findings have actually changed the dietary guidelines, which is now putting more emphasis on consuming healthy type of fat instead of reducing total amount of fat in the diet. But the controversy regarding saturated fat has continued. You may have seen those headlines, eat butter, butter is back, is butter really back? So the main issue is what's the role of saturated fat in our diet? The major issue in this debate is what is saturated fat compared to? So what is the comparison macronutrient? So in this study, we found that if you replace saturated fat with good fats, like monounsaturated fat or polyunsaturated fat, there is a significant benefit in terms of cardiovascular disease. However, if you replace saturated fat with refined carbohydrates and the sugar, then there is no benefit at all. So it means that reducing saturated fat and eat more white bread and refined starches and drinking more Coke, that's not good for you at all. So this, I think, is very important when we make public health recommendations. We always have to talk about the replacement nutrients or replacement food. And this type of findings was, again, underscore the importance of type of fats, type of carbohydrates, or the quality of fat and quality of carbohydrates in our diet. As Mathilde mentioned earlier, there has been extremely consistent evidence on the harmful effects of sugary beverages on a wide range of health outcomes. In our cohorts, we have found robust and consistent evidence that higher consumption of sugar-sweetened beverages is associated with increased risk of obesity, type 2 diabetes, cardiovascular disease, some cancers, and also premature deaths. On the other hand, higher consumption of tea and coffee, in particular, associated with lower risk of chronic disease. And the findings on artificial sweetened beverages are interesting because this is a beverage which is much more difficult to study than sugary beverages because of the reverse causation issue, confounding, and other methodological issues. So in our cohorts, we found that very high consumption of artificial sweetened beverages, 2 or more servings per day, is associated with increased risk of cardiovascular mortality. But relatively low consumption seems to be okay. But again, this is a still evolving area and require much more extensive, not just epidemiological studies, but also experimental studies. Recently, we published a paper on 24-hour urinary sodium and potassium excretion and cardiovascular disease. And this is a very important, but also controversial topic because several studies have found a J-shaped or U-shaped relationship between sodium consumption and the risk of cardiovascular disease. The lowest risk was actually found among people who consume 4,000 milligram of sodium per day. This is much higher than what WHO has recommended, which is around 2,000 milligram per day. But the main methodological problem with those studies is that they use single spot urine samples to measure sodium intake. We know that this type of measurement is susceptible to not just the random measurement errors, but also biases. What we did is to use multiple 24-hour urine samples, which is a gold standard to measure sodium and potassium intakes. What we found is that there is a dose-response relationship between increasing 24-hour urinary sodium excretion and the risk of cardiovascular disease. There is no evidence of J-shaped relationship or U-shaped relationship. And then for potassium, as you can see that there is also a dose-response relationship between increased 24-hour urinary potassium excretion and decreased risk of cardiovascular disease. So this kind of studies underscore the importance of using valid biomarkers as a surrogate for dietary intakes and assess the relationship between those biomarkers and the risk of chronic disease. So these findings, I think, have very important public health recommendations, and because they support the current American Heart Association recommendation, the WHO recommendation, and also the USDA dietary guidelines that reducing sodium intake is an important public health strategy to reduce risk of cardiovascular disease. We have conducted extensive research on consumption of animal products, especially red meat and processed meat, and the risk of chronic diseases. The evidence has been very consistent. High consumption of red meat, especially processed red meat, is an important risk factor for diabetes, cardiovascular disease, some cancers, and also premature deaths. And cooking methods also matters because in our analysis, we found that high temperature cooking, such as grilling or barbecuing of meat, especially red meat, is associated with increased risk of diabetes and other chronic diseases, independent of the amount of red meat consumption. And also, when we look at the different types of proteins, typically animal protein, higher animal protein tends to be associated with increased risk of chronic diseases and premature deaths. On the other hand, higher consumption of plant protein is beneficial for the prevention of chronic diseases and mortality. Recently, we look at the relationship between gut microbial metabolite, TMAO, and the risk of cardiovascular disease. What we found is that increasing TMAO in the blood is associated with increased risk of cardiovascular disease. So in the US population, TMAO is significantly correlated with red meat intake because TMA, when people consume red meat, the components of red meat, such as collagen, are metabolized by the bacteria in the gut, which need to increase production of TMAO. And so this type of studies provide further evidence that higher consumption of red meat is an important risk factor for cardiovascular disease. On the other hand, higher consumption of minimally processed plant foods has been consistently associated with lower risk of chronic diseases. So in this study, we found regular consumption of nuts, including tree nuts and peanuts, is associated with lower mortality and increased longevity. And then fruits and vegetables, especially the specific type of fruits and vegetables, are associated with lower mortality. Even though olive oil consumption is relatively low in the US population compared to that in the European population, the consumption has increased dramatically in the US population in the last 2 to 3 decades. So what we found is that even moderate consumption of olive oil 3 to 4 times per week is associated with lower risk of cardiovascular disease. And then in terms of whole grains, what we found is that higher consumption of whole grains, including different types of whole grains such as brown rice, barley, oats, is associated with lower risk of chronic diseases and mortality. But the consumption of whole grains in the US is very low. It's about 1 serving per day, which is much lower than that recommended by the US dietary guidelines, which is about 3 servings per day. And then again, the most remarkable finding from our cohorts and also other studies is the beneficial effects of regular coffee consumption on the risk of diabetes, cardiovascular disease, and mortality. So, so far, at least 3 dozens of large cohort studies around the world have found an inverse association between regular consumption of coffee and the risk of type 2 diabetes, CVD, and mortality. Certainly it's good news for most of you or for all of you here. But the biological mechanisms, I think, are extremely interesting because we know coffee contains not just caffeine, but also hundreds, perhaps thousands of bioactive compounds, especially polyphenols. So I think this is area warrants further intervention studies, mechanistic studies to better understand the biological mechanisms. Of course, it's not a good idea to add a lot of sugar or cream to your coffee. That's what people do when they go to Starbucks coffee, which is not a real coffee anymore. It's basically sugary or milk drinks. Another important finding is that not all plant-based diets are healthy. So plant-based diets have been widely recommended to reduce risk of chronic disease and also improve environmental sustainability. But what we found is that plant-based diets rich in minimally processed plant foods, such as fruits and vegetables, whole grains, nuts, vegetable oils, coffee, or tea, are beneficial. On the other hand, plant-based diets rich in refined grains, potatoes, fries, sugary beverages, sweets, are actually associated with increased risk of chronic diseases. This kind of diet is actually worse than animal food-based diets. But in this kind of toxic food environment, it's so easy to consume unhealthy plant-based diet. I mean, if you go to college campuses, some people say, oh, I'm eating veggie pizza, I have a Diet Coke, but that's maybe worse than the regular diets which contain relatively high amount of animal products. So again, this kind of findings underscore the quality of plant foods in our diet. As the previous speaker just mentioned, ultra-processed foods, or UPFs, have received a lot of attention. And in our cohorts, working with Carlos, we found that higher consumption of UPFs is associated with diabetes, colon polyps, and also maternal consumption UPFs is associated with subsequent risk of offspring, overweight, obesity. So clearly, as an overall category, UPFs are bad. But what's interesting in our cohort is that there is a heterogeneity in the effects of UPFs. So some of the foods which are classified as UPFs are actually not bad or even beneficial. For example, we found that whole wheat bread, some brands of cold breakfast cereals, or even some brands of yogurt, which are classified as UPFs, they are associated with lower risk of obesity and type 2 diabetes. So even though overall category of UPFs is clearly bad, but we have to pay attention to the heterogeneity or the variability of different categories of UPFs. Now I'm going to switch gear to talk about personalized nutrition and precision nutrition because it's a very hot topic. Last year, NIH funded a huge precision nutrition study, $170 million. So this is the largest single nutrition study funded by NIH, $170 million. And the rationale for personalized nutrition or precision nutrition has been around for years or even decades because we know one size doesn't fit all. Different individuals have different metabolic responses to the same food or the same diet. And according to the 2020-2030 strategic plan for NIH nutrition research, precision nutrition is defined as a very broad framework that canvasses a wide array of features, including your genes, your microbiome, your metabolome, but also your physical activity, your food environment, and your circadian rhythm, and so on and so forth. So this concept is much broader than the traditional concept of personalized nutrition or nutrient genomics, which is aimed to tailor nutrition strategies to individual characteristics such as genetics, genetic variants or gut microbiome. So precision nutrition recognizes that humans are inherently different from one another. So when, why, and how we eat is likely as important as what we eat. So this is a precision nutrition pyramid. At the bottom of the pyramid, so everyone should get a general diet, healthy diet advice. I mean, eat more fruits and vegetables, more whole grains, reduce consumption of processed foods. But on top of that, specific diet recommendations can be made based on individuals' health conditions and food preferences. So you can recommend Mediterranean diet, vegetarian diet, or ketogenic diet, or intermittent fasting to different people with different health conditions and food and cultural preferences. And then on top of that, molecular nutrition advice can be made based on nutrition omics data and also our understanding biological mechanisms. So this is the ultimate goal of precision nutrition or personalized nutrition. This is where most of the exciting research is happening right now. In the past 15 years, we have been collaborating with Collège at the Broad Institute to conduct large-scale, high-throughput metabolomics profiling of participants in our cohorts and also in the PREDIMED trial. The platform is based on the LCMS method, and it can detect more than 700 non-metabolites and thousands of non-metabolites. This platform is highly reliable, very robust, and reproducible. So far, we have analyzed more than 20,000 participants in our cohorts and more than 3,000 participants in the PREDIMED trial. So metabolomics is one of the precision nutrition tools, and it has become, I think, very important for precision nutrition research, for nutrition epi research. But in general, precision nutrition has three utilities in terms of nutrition research and practice. The first one is to improve dietary assessment and compliance. The second one is better understand biological mechanisms which can enhance the causality interpretation and strengthen current or future dietary recommendations. And the third one is to offer more effective personalized nutrition strategies to improve health outcomes. So using the metabolomics technology and also using the combination of feeding studies and observational analysis, many plasma or urinary metabolites have been identified to reflect intakes of certain foods, beverages, and eating patterns. So theoretically, those metabolites can be used as objective biomarkers of dietary intakes. One caveat is that many of those metabolites are not very specific to certain foods or food groups. As an example, plasma levels of TMAO is correlated with red meat intake, as I mentioned earlier, but it's also correlated with fish intake because you can consume TMAO directly from fish. So those are two different ways that you have elevated TMAO levels. So if you just measure plasma levels of TMAO without knowing the dietary habits of the individuals, you wouldn't be able to tell whether this person is a red meat consumer or a regular fish consumer. Recently, we published a study in which we identified a metabolic signature that can distinguish healthy versus unhealthy plant-based diet. So as I mentioned earlier, a healthy plant-based diet is beneficial, but unhealthy plant-based diet is actually worse than animal food-based diet. And we developed a signature that can distinguish those two dietary patterns, and also this signature was strongly associated with lower risk of diabetes, independent of BMI, and other diabetes risk factors. So this signature can be potentially used as a biomarker for plant-based diet and can be used to monitor adherence to dietary interventions in feeding studies or in dietary intervention studies. And using similar method, we also identified a signature for adherence to Mediterranean diet. So the signature was initially identified in the PREDIMED trial, which is a large primary prevention trial of cardiovascular disease using Mediterranean diet supplemented with extra virgin olive oil or mixed nuts. And this signature consisted of 67 plasma metabolites. And when we validated this signature in our 3 large cohorts in the US, and then this signature predicted a lower risk of cardiovascular disease in both the Spanish cohort and also the US cohort. And then to demonstrate the causality of the signature and the risk of CVD, we conducted Mendelian randomization analysis with genetic variants that are correlated with the signature. We found that the genetically inferred metabolic signature is also associated with lower risk of CVD. So this is one of the examples how nutrition epidemiology can be combined with multi-omics studies to not just identify novel biomarkers, but also enhance the causal interpretations between the nutrition exposure and the risk of chronic diseases. And again, this type of analysis can help us better understand the biological mechanisms at the molecular level, and also can help us better quantify individuals' variability in response to the dietary interventions. Just want to give a quick example. We have been interested in the metabolism of tryptophan and the risk of chronic disease. So tryptophan, as you know, is an essential amino acid, and it plays a very important role in immune function, brain function, and also obesity and diabetes. It's primarily metabolized through 2 pathways: the host metabolism pathway, which produce chreonine and the chreonine-related metabolites. The other pathway is microbial or gut metabolism pathway, which produce different type of metabolites like IPA or indole-3 propionic acids. And those metabolites can serve as signals for insulin resistance, for chronic inflammation, and also for other metabolic mechanisms. And those metabolism pathways of tryptophan can be influenced by multiple factors, including genetics, diet, and also gut microbiome composition. So what we did is to use a large cohort of US Hispanics as a discovery cohort. So this cohort includes 16,000 US Hispanics or Latinos in which very detailed data on diet and also multi-omics and data available. And then using several other US cohorts as a replication cohort, including the ERIC cohort, Framingham cohort, PREDIMED, and the Women's Health Initiative. So this analysis include multi-ethnic comparisons and also multi-ethnic replications. So I don't have time to get into the details. I just want to highlight a few main takeaways from this very complex analysis. First, the host tryptophan and its creatinine-related pathway metabolites showed a positive association with type 2 diabetes, where the microbial pathway metabolites like IPA showed inverse association. So clearly, the microbial pathway metabolites are beneficial in terms of improving metabolic profile, reducing risk of type 2 diabetes. What's really interesting is that plant-based fiber-rich diet were associated with increased beneficial metabolite, like IPA. On the other hand, a diet high in animal protein was associated with increased creatinine metabolites. Those are harmful metabolites. So this kind of findings can help us explain why higher animal protein is associated with increased risk of diabetes and other chronic diseases, whereas a healthy plant-based diet, fiber-rich diet, associated with lower risk of those diseases. And we also found that the effects of fiber-rich diet on beneficial metabolites are partially mediated through gut microbiome composition. So again, this is important finding because we know that a fiber-rich diet can help to improve gut microbiome, and a healthy microbiome can also shift the metabolism of tryptophan from the harmful pathway to a more beneficial pathway. So in recent years, microbiota targeted interventions have become a promising personalized nutrition strategy. And those strategies include whole diet intervention, increasing fermented food consumption, prebiotic, probiotic, and symbiotic interventions. There is increasing evidence that microbiome composition can modulate the effects of diet on cardiometabolic health. In this study, we found that Mediterranean dietary patterns associated with microbial species involved in plant polysaccharide degradation and the short-chain fatty acid production, and also the effects of the Mediterranean diet on cardiometabolic health. It's modified by certain bacteria species in the gut. And similarly, we found that the effects of red meat consumption on TMAO are modified by the abundance of TMAO producers in the gut. So the increased TMAO production among those who consume high amount of red meat is only observed among those who carry large amount of TMAO producers. Similarly, we found the interaction between consumption of lignans and the gut microbiome composition and the production of this lignan metabolite called enterolactone. Enterolactone has been found to be associated with reduced insulin resistance and type 2 diabetes risk. Again, the relationship between dietary lignan and the production of enterolactone is modified by the abundance of enterolactone species in the gut. So those examples illustrate that the production of certain harmful or beneficial metabolites are not just determined by our diet, but also determined by the gut microbiome. So that's underscore the complexity or the interplays between our diet, our genes, and the microbiome on cardiometabolic disease risk. And in recent years, several computer algorithms or AIs have been developed to predict individual responses to diet. And also those computer algorithms or AI tools have been used to design personalized diet to lower glycemic response and improve metabolic health. But this is still in the early stage because those algorithms need to be validated in external populations, and also the cost of doing this type of personalized nutrition consultations is still very high. So where do we stand in terms of precision nutrition in nutrition research and practice? In terms of dietary assessment, I think this Omics tools, especially metabolomics, hold promises for novel biomarker discovery for food intakes and dietary patterns, but they are complementary to rather than a replacement for traditional nutrition biomarkers and self-reported dietary assessment tools. Even now, we have very exciting metabolomics biomarkers for certain foods and dietary patterns, but this doesn't mean we will get rid of self-reported tools like 24-hour recalls or validated FFQs. And I think those kind of tools are most exciting in terms of understanding biological mechanisms. And as I mentioned earlier, that can help us shift from the black box epidemiology paradigm to systems epidemiology paradigm. However, although new mechanisms may be discovered, clinical translations are challenging and will take time. In terms of personalized nutrition advice, AI-based tools are still in the early stage. I don't think they are ready for prime time yet. Commercial products typically outpace the scientific evidence. They are more useful for disease management than for prevention. And we also should be aware that this kind of tools may actually widen health disparities. So that's why we need to find a right balance between public health approaches and the precision nutrition approaches. So right now, precision nutrition is a very hot topic. As I mentioned, NIH spent $100 million last year only on one single precision nutrition project. But historically, public health nutrition research has been underfunded. So I think it's very important for us to strike a right balance between public health nutrition and precision nutrition. And nutrition epidemiological studies have already made important impact on dietary guidelines and nutrition policies in the US and worldwide. For example, the US dietary guidelines now put more emphasis on type of fats and have removed the 30% upper limit on total fat. So it's not emphasizing no-fat diet anymore. It's actually emphasizing quality of fat. Now it recommends replacing healthy fats with unhealthy fats with healthy fats rather than carbohydrates. And as you know, the FDA mandated trans fat labeling in 2006 and then banned trans fat in the US food supply in 2018. And the FDA has also revised the nutrition facts label several years ago and added a new line on the amount of added sugar on the nutrition facts label. Before that, we have a line on total amount of sugar, which is not very meaningful. And now, currently, the revised nutrition facts labels include not just total amount of sugar, but also, more importantly, added sugar on the labels. And then the dietary guidelines have put more focus on healthy eating patterns instead of individual nutrient. So commonly asked the question is healthy diet for humans also beneficial for the health of planet? This is a recent published paper from our cohorts looking at health and environmental impacts of plant-based dietary patterns. As you can see that as the diet score improves, there is a parallel decline in the risk of cardiovascular incidents and also in greenhouse gas emissions. So clearly, a healthy plant-based diet is not just beneficial for human health, but also beneficial for the environment. So that's why we need a systems approach to integrate the tools and the strategies from precision nutrition, from public health nutrition, and from planetary nutrition to improve personal health, population health, and planetary health. Our personal health depends on the health of populations or communities, and our human health depends on the health of the planet. So that's why we have to use a holistic approach to improve both human health and the health of the planet. So I'm going to stop here. Thank you very much for your attention.